Hello everyone. We are happy to present you VAR Decrypt. To start, user has two options: running a new analysis or reloading previously processed data. VAR Decrypt can handle somatic and germline variants. VAR Decrypt requires two input files. User can find an example of these files in our Git repository. After selecting input files, the user can click on the Load button to import samples data. As we are processing somatic variants in this tutorial, we will check Somatic Variants tab to visualize imported data. On this page, users can select different parameters such as filtering imported data to keep genes annotated in one of the three proposed databases: COSMIC, OncoKB, and NCGDB. Moreover, users can filter data to keep genes with less than 4 mutations in 15% of the patient cohort, for example. After selecting suitable parameters, the user can click on the Process button to start the processing step. Depending on the size of the data and the number of patients, samples, this step may take varying amounts of time. In this example, it takes 5 minutes to process data from 59 samples containing about 2,000 single genes. By the end of the processing step, users can explore the different results. Let's start by checking the plots in General Statistics tab. In this tab, the user has 6 useful plots. 1. Somatic Variant Summary, which provides users with several information about patient's cohort. 2. Mutation Oncoplot, which provides users with an additional set of information about the top mutated genes and the type of mutation by patient. Moreover, it is possible to check mutation information for particular genes by typing their name in Select Some Genes box. 3. Transition, Transversions, which shows the result of the classification of SNPs into transition or transversions. 4. Mutations, TCGA, which is a comparison of burden mutations in samples data against 33 TCGA cohorts loaded from the Cancer Genome Atlas. 5. Somatic Interactions, which is a function from MAFTools package. It performs pairwise Fisher's exact test to detect mutually exclusive or co-occurring mutations. Color code shows the type of the interaction. Six. Drug-gene interactions. This plot shows the drug-gene interaction among the mutated genes in the patient's cohort. Now let's move to Enrichment tab. Here we can see the enrichment results using several databases. Users can choose between 3 visualization plots: Q-value bar plot, enrichment factor, and cluster tree. VAR Decrypt incorporates the following enrichment analyses: disease ontology, reactome pathway, biological process, molecular function, cellular component, gene disease associations, gene cancer associations, KEGG pathway, and Wiki pathways enrichment. For each enrichment analysis, user can download the full enrichment results table in CSV and in Excel format, and he can of course download the plot in PDF format. In Enrichment of Known Oncogenic Signaling Pathways tab, user can explore the enrichments for a known oncogenic signaling pathways according to the TCGA cohort's data. Users can get more detailed information about genes involved from each patient by simply clicking on a displayed pathway on the right part of the plot. Tumor suppressor genes are colored in red, and oncogenes are in blue. In Amino Acid Changes tab, the users can see precisely where the variants, mutations are located on the coding gene's corresponding protein sequence and how they are distributed in the cohort, in order to detect mutational hotspots for instance or functional domain alterations. To do so, users need to write the name of the desired gene in the Select a Gene box. Finally, var decrypt implements a function to cluster the samples according to the type of mutation for a given list of genes. User needs to select the number of cluster and type the name of genes in Select Some Genes box. Then click on Submit. Mutations have the same weight option, allow user to ignore the difference in the number of mutations by gene between the patients. Thank you for watching.